All right. Good afternoon, everyone. It is 1 p.m. on the east coast of the US, so it is C-Star time. My name is Sigurd Christensen, and I'm happy to welcome our in-person and online audience to today's presentation. Before hearing from today's speaker, I want to alert you to our talk in 2 weeks' time, which will be given by Dr. Greg Hickok from the University of California, Irvine, who is well known to most of our audience. The title of his talk is Wired for Words, the Neural Architecture of Language and its Sensory Motor Foundation. So make sure to tune back in 2 weeks' time. Today, however, it gives me great pleasure to welcome Dr. Galit Agmon. Dr. Agmon is a senior lecturer at the Department of Linguistics and a member of the Gonda Multidisciplinary Brain Research Center at Bar-Ilan University. Her work is informed by a background in linguistics and cognitive sciences, and she's completed her PhD at the Hebrew University of Jerusalem in 2018 and a postdoctoral fellowship at Bar-Ilan University in 2021 and at the Frontotemporal Degeneration Center at the University of Pennsylvania in 2024, before settling into her current position at Bar-Ilan University. And today, she'll share some of her work on spontaneous speech as a window into the Syntax-Prosody interface. And please feel free to write your questions in the chat box during her talk for Dr. Agmon to answer at the end of her talk. And with that, I'll give the floor to you, Galit.
Thank you so much. I'm so excited to be here and to talk with you today. The name of my talk is Spontaneous Speech as a Window into the Syntax-Prosody interface. So when Zook was first contacting me and asked me if I was willing to give a talk, I told him, well, I've been working on aphasia, and I'm going to present you that data today as well, automated speech analysis of people with primary progressive aphasia. But now I'm mostly doing cognitive research, which I will also present today, but it is very much related. And the way it is related is through the challenges of working on spontaneous speech, and I'll focus today on syntactic boundaries. So as I said, I'll start with automated speech analysis, specifically syntactic complexity analysis of people with primary progressive aphasia. The challenge there was that in order to analyze syntax, I needed to have syntactic units. And the syntactic units is what people, call sentences, which may be a charged word, but I will use that throughout the talk. And that is not it's not that easy to define what a sentence is. And this idea kind of resonated with me when I started working at Bar-Ilan University on the cognitive and neural processing of syntactic boundaries, because if this was a challenge for me as a kind of engineering challenge, it is also a challenge for the brain. And this leads me to what I am doing today, which I'll present at the end, on the quantifying the prosody syntax alignment. So this is very preliminary. This is just one grant, 5-year grant. I'm working on it right now. So any feedback is actually very helpful because these are ideas that I'm processing, working on, and I'd be very happy to hear what your thoughts and ideas about this. So the first part of the talk will focus on pathological speech. The second part will focus on neurotypical speech from a cognitive perspective, and then I'll describe what I'm doing right now, kind of, having both backgrounds in mind. So we'll start with syntactic speech analysis, which is what I did as a postdoctoral fellow at the Penn Frontotemporal Degeneration Center. So when I started my postdoc, I must admit I did not know anything about PPA, PPA is a subtype of FTD, frontotemporal dementia. I never heard about that. And I have the impression that now it is more known, probably because of Bruce Willis, who's made it more, people hear more about it, and his family is really doing a great job raising awareness to FTD and to this very terrible disease. Um, a month ago, I actually ran across on BBC um, this little piece of news about the youngest ever person who was diagnosed with FTD, who passed right before Christmas. This was a month ago. He was diagnosed when he was 23, passed when he was 24. So I don't know, maybe I'm biased, but I feel that people write more about this, but I don't want to assume that, everyone knows what FTD is. I did not know what FTD is. So FTD, frontotemporal dementia, as its name implies, neurodegeneration that starts, or the focus of the initial atrophy is in the frontal and temporal lobes. It's considered an early onset dementia, as the example that I just showed you, which is a very rare example, but it usually starts on average before the age of 60. About 7 years, if I'm not mistaken, between diagnosis and death. It's caused by misfolded proteins in the neurons that aggregate and cause neuronal death that spreads throughout the brain. It is progressive, it has no cure, and it's a spectrum of conditions. And as we all know, the language network is mostly in frontal and temporal regions. It is not surprising that this kind of condition also affects language. And the language specific or language related FTD subtypes are referred to as primary progressive aphasia, PPA. We can subtype these as well into the non-fluent agrammatic primary progressive aphasia, NAPPA, which starts mostly in the left inferior frontal gyrus, in the frontal lobe, and the semantic variant SVPPA, which starts in temporal regions. There is also another subtype, LVPPA, but this is mostly associated with Alzheimer's pathology, but symptomatically, they start off or may exhibit similar symptoms. So what are the speech symptoms of PPA? Again, this is a sort of quick background. The speech samples that I was analyzing, me as part of the speech team at the FTD center at the UPenn, came from mostly from the cookie theft picture description. Now, I have a lot of things to say about the cookie theft picture description, and I'm happy to talk about this at the end, but this is really part of the battery that is used for decades in the diagnosis of aphasia. And here are just like two speech samples of NAPPA and SVPPA, which I'm going to focus on. Let's listen to the first one, NAPPA.
Go ahead. There is a boy and a girl. The boy is taking cookies out of the cupboard. He's going to fall down. The mother is at the sink in the kitchen.
Okay, so we heard, I think the most prominent feature is very halted and effortful speech. What you may have also noticed, but maybe not that easy to notice in this very short sample, there is also grammatical degradation. They have grammatical errors, simplification in their speech, omission of function words, lack of inflection. So this is parallels to Broca's aphasia because of maybe the region of the atrophy. And since this is progressive, gradually they will lose effective communication. So let's now hear a sample of SVPPA.
A man is standing on something, and a woman is there with him, and a mother is holding something near there. They're all in the house.
So this was an SVPPA patient describing the cookie theft picture description. There were many naming difficulties. So although fluency seemed to be intact, there were many pauses trying to find the suitable words to describe the picture. And often these words were too abstract, a bit, associated with, but not exactly describing, like man instead of boy, thing, something. And this is really the hallmark of SVPPA, of losing the content of words. So speech becomes gradually empty. This is not just language. They lose their world knowledge until they are really disabled and cannot function at the end. But at the very beginning, these changes in speech are more subtle than this. So at the speech team at FTD, when I came there, Murray Grossman was at the time the head of the FTD Center. He wanted me to analyze the syntax of these patients from the speech samples that we had. And at the time, it was mostly the acoustic features or lexical features that were analyzed, and syntax was a bit more complex. It was a bit more complicated. So that's what I was working on as a postdoc. So what I did, I extracted from the speech sample describing the cookie theft picture syntactic features such as relative clauses, center embedding, noun modifications, and so on. I'm not going to go into much detail describing exactly how I did this. I'm happy to answer this at the end, but all of these features that I extracted from the speech per patient, I combined using statistical methods into a score that was supposed to reflect the syntactic complexity of the speech of that patient or that person. So this is this is a speech biomarker that was supposed to help us to screen and monitor the disease, to subtype the aphasias and to be able to monitor the disease. And the hypothesis here was that the NIPPA patients would be the most sensitive to this syntactic complexity score. And this was really nice because it was pretty successful. So first of all, validating this on healthy participants. So these are participants, younger and older adults without PPA. And what I showed here was that the older people in their speech, again describing the cookie theft picture, had a lower score in their syntactic complexity, which aligns with, first of all, evidence from studies that we know about decline in syntactic complexity or syntactic structures that are exhibited in the speech of older adults. But also this was manually validated. It was a lot of work. Now turning into the PPA patients, the controls here are age matched. So these are the same old on the left. So comparing to the healthy controls, age-matched healthy controls, the PPA patients had lower scores, but the NAPPA patients had the lowest scores. So it stood out that the NAPPA patients scored the lowest on syntactic complexity. And the NAPPA patients, those for whom we had longitudinal data, were also the only ones to decline over time. So unlike the LVPPA or the SVPPA patients that were, they were low, but they were pretty consistent in their syntactic complexity. The decline in NAPPA was significant. And this was a biological validation. For some of the patients, we had measurements of their neurofilament light chain level and their CSF. So NFL it supports the structure of the axon. And when there's neuronal damage, it is secreted into the CSF. So high levels in CSF are an indicator or a biomarker of neuronal atrophy. But of course, we don't know where the atrophy is. The assumption is that in NAPPA patients, since the focus is mostly in frontal regions that are related to grammar, higher levels of NFL and CSF should negatively correlate with their syntactic complexity score. And this is indeed what we found. This correlation is statistically significant, and we did not find that same association with the SV PPA patients. So this was nice. This was my postdoc, 3 years in Philadelphia, 3 great years in Philadelphia. And it wasn't that easy. I mean, I just presented it just like that, as if, hey, I just extracted these features and here is a score. But it wasn't easy. And the reason that it wasn't easy is because speech is disfluent. And it's not just because we're talking about patients. Speech in general is disfluent. And disfluencies, by definition, do not have a position in the syntactic tree. They need to be cleaned out in order to have a clean representation of syntax from which then I can extract these syntactic features. Now, up until now, I spoke about production in pathological speech, but now we're going to move to speech in healthy people because, as I said in the introduction, the work on automated speech analysis really resonated when I was working on cognitive analysis and speech comprehension. So in order to demonstrate how disfluent spontaneous speech is in healthy people in neurotypical populations, I have here Quentin Tarantino. There's nothing, particularly special about Quentin Tarantino, but when I was thinking about these things, think at the time he got married with Daniela Pick and then moved to Tel Aviv. So I read the tabloids and it was just there. So I was just searching for something. It could have been anyone else, but this is him promoting Inglourious Basterds in 2009, just answering questions spontaneously. Let's just hear him. And then I'll say a few things.
Part of the fun experience of going around the world and promoting my movies is actually I try to add a couple new countries that I've never been to each time. All right, I go on the tour.
Okay, that's it. Short, easy to understand. I don't think anyone who hears this is thinking to themselves, "Oh, what's wrong with Quentin Tarantino?" Because we all talk like that. We all understand people who talk like that. But let's just try to write down what he said word by word and then take a look at it again.
Part of the fun experience of going around the world and promoting my movies is actually I try to add a couple new countries that I've never been to each time. All right, I go on the tour.
Okay, so what I like to do here usually is to just let people to have a minute and read it. And it's a very weird experience to read it because it's difficult. And when we were hearing it, it was very easy to understand. But actually what we have here, what our brain had to deal with when we heard Quentin talking at the first video that I showed you, we had to deal with word repetitions, right? And we had to deal with fillers, non-lexical fillers like and that we use a lot in everyday speech. We also had to deal with lexical fillers. These are words that have lexical entries, but here they play no role in any semantic or syntactic function in the building up the meaning of the sentence. So we have here, all right. And we also have restarts, which also happens a lot in spontaneous speech. In the middle of the sentence, we just decide to start a new sentence, and everyone is okay with that. Right? This is something that is very natural for us in everyday communication. It is easy to understand. However, not however, it is easy to understand because what we think that we hear, if I had to now ask every one of you to write down what he said, you would probably write down something more similar to what is on the right. And it's not just because, you're trying to polish disfluent speech. This is usually what people remember. They very easily forget or do not pay attention to these disfluencies, right? These word repetitions and fillers and restarts. So if it's something that we do, right, that it means that this is something that our brain has to do. And this is a puzzle. And this is a puzzle because for decades of research in psycholinguistics and neurolinguistics have used sentences, right? A representation of a sentence, right? S goes to NP and VP, noun phrase and verb phrase. There's a lot of evidence of, a syntactic representation that our cognitive system is processing. And if we do automated speech analysis, it's not even our brain. We need this representation to extract syntactic features. So if we want to analyze that speech, we need to have that syntactic representation. And it works well if you have a planned, controlled experiment. But if you want to analyze or study speech as we use it in everyday life, what we have is something which is far from this, clean sentence.
Part of the fun experience of going around the world and promoting my movement.
Something that sounds like this. So the question is, how do we get from the messy, disfluent input on the left to the clean representation, clean and segmented representation, right? Something that I didn't mention is we also need to have a sentence in order to have this nice representation. So what we need to do, we as, if you want to be like an engineering problem and analyze speech using NLP tools, or if you want to understand what the brain does, right? This is like from both sides. We need to remove the disfluencies. And we need to segment this continuous stream of input into sentences in order to obtain the representation of S goes to NP and VP. And this is something that I called, speech pre-processing. It's pre-processing, in the data analysis sense, but also something that I believe that our brain has to do somewhere, somehow. And the nice thing is that we have evidence for that. So the evidence comes from work that I did at Bar-Ilan University in collaboration with Ilana Tzion Golombik. And we showed evidence that I'm focusing now on removing disfluencies, that our brain is sensitive to disfluencies and suppresses their representation. And this was published in the Neurobiology of Language 2 years ago. And this is what we see here in this is from EEG, okay? And green, non-fillers is actual words. When I say words as opposed to fillers, I mean words that have a syntactic position and play a role in the semantic meaning at the end, as opposed to fillers in gray here, which do not. The response is kind of flat. So this is a suppression of disfluencies that our brain does. Now, it's worth maybe taking a few minutes to say how exactly we measured this. So as I mentioned, this was EEG. We had 20 participants who listened to
שהפרצות תהיה משהו
spontaneous
הרבה,
speech.
כאילו יותר מאשר צחוקים ובלאגן בפייסבוק.
So this was in Hebrew, and although most of you don't understand Hebrew, it's easy to recognize spontaneous speech in any language when you hear it. They heard this 6 minute of spontaneous speech, their brain signals were recorded with EEG. And the analysis here is not a typical EEG analysis. Okay, we did not just average all the responses time locked to fillers. What we did here is something a bit different. So if you have you have a continuous input, the acoustic signal is a continuous input, and a continuous output, the neural response, mathematically, you can ask, is there a function that explains the transformation from one to the other, right? So it's like as if every instantaneous point in time in the acoustic signal causes some typical average response across the whole signal. We're not talking about being time locked to a specific word. We're talking about a continuous response, or you can think about it as a filter. So this is, simple convolution, right? In a way, I just didn't want to assume that everyone knows what convolution is. And this gives us the response function, the instantaneous response function across the whole input, which is called the temporal response function, or TRF. So for those who know fMRI, this is kind of analogous to the HRF in fMRI, only that the TRF is extracted and not assumed. And so what we see on the bottom is a TRF response, okay? But we have two separate TRF responses because what we did is that we segmented the acoustic input into speech of actual words in green and speech of disfluencies in gray. And this TRF, this auditory TRF response it's worth mentioning. It has been studied in EEG and MEG, and it was shown to be modulated by attention. So it's suppressed when you don't attend to, it's usually like the attended speaker, the non-attended speaker. So showing here a modulation of this auditory TRF by whether the word, the utterance is a filler or not a filler, suggests to me that we have some early auditory filter that, again, when I say early, I'm talking about auditory regions. It detects the disfluencies and suppresses them, flattens the response to them before moving on, let's say, to more like downstream linguistic or higher level linguistic analyses. We also had a bunch of controls that I don't have time to go into, but again, asked me at the end. We looked at the duration of the words, the volume of the words, the information level of the words, giving it, very briefly. It really was about whether it was a filler or not a filler, not about how much, for example, how much information that word conveyed. For example, when we look at content versus function words. But again, feel free to ask me at the end. So this is very nice, like an initial evidence for a filtering mechanism, an acoustic auditory filtering mechanism that cleans the messy input before further analysis. But if we're talking about syntactic analysis, we need a sentence. So from the same experiment, I marked the syntactic boundaries, not necessarily of sentences, but major syntactic boundaries. And we looked at what happens on syntactic boundaries to the TRF. And we saw that the TRF was modulated by syntactic boundary. If the word was a closing word, a word that closed a major syntactic phrase, the TRF was, the latency was longer, the peak was higher. There are many interpretations to give to this. This really requires further investigation, but this is, again, evidence for an early sensitivity to pre-processing the speech, the messiness of speech, before we can even consider syntactic analysis and semantic interpretation. But then, what is a sentence, right? If I segmented it into major syntactic boundaries, or I need to segment speech into sentences in order to analyze it, or our brain has to segment the continuous stream of speech in order to analyze it. But what is a sentence, really, right? It's not an easy question to answer, although we all feel that we know what it's a sentence. This is a sentence, right? Linguists know what a sentence is. A sentence is S, right? The label S, and then you can analyze it, and that's a sentence. So, I mean, it is more complex than that. You can ask, for example, is a conjunction of two sentences one sentence or two sentences in the brain, for example? And I worked with a very specific definition of a sentence. So conjunctions, conjoined sentences are two separate sentences. Intuitively, I hope you can all have a feel of how or why conjunctions are not do not make one huge sentence, but rather two sentences. People use and all the time in continuous speech, maybe almost as a filler. So this is a theoretical question, but also, again, from the brain's perspective, if the brain really represents the syntax in order to extract the semantic interpretation, which is, again, what linguists and psycholinguists, most of them, have been working based on this assumption for decades, then how does our brain know when one sentence ends and another sentence begins? How does it do that? Because what it gets is just, an endless string of words. How do you know what I'm saying right now? I just restarted a sentence. I don't know how many of you have just noticed that I did that. It's so natural. But even if I didn't restart a sentence, when I end a sentence, how do you know I'm not continuing it, but rather starting a new one? So this is really a puzzle, although many people just say, or may just say very naturally, well, it's speech. It's not reading or writing, right? In speech, we have something, a very useful tool, which is called prosody. And prosody, right? The musical colors of speech, the change in intonation, the duration, the rate, the volume, intensity, and all of that is a very significant cue. So when I started reading prosodic literature, I was surprised to discover how much this world is kind of encapsulated within itself. So of course, everyone agrees and knows this is not, no one is falling off the chair. Everyone knows that prosody is very helpful and supports syntactic segmentation. But in the world of prosodic analysis, they talk about intonation units. And this is independent of syntax. There is some prosodic flow, and then there is some discontinuity and reset. And that's the point where a new intonation unit starts. And it may very well be aligned with syntactic boundaries, but it has its own independent definition, or features, let's say, not definition. So what are the features of prosodic breaks, right? So if we have a string of words, very often at a prosodic break, we will have a pause, right? If we look at the duration of these words, how long each word is relative to normalized, let's say, by the number of syllables, very often the word right before the pause at the break is much longer than what it would have been if it wasn't right before a pause. And looking at pitch, again, this is on average, very typical. There is some declination in pitch, also in volume intensity. People start high and then they go low. And it makes sense. At the pause, you need to take air, you need to start again, and then you start with full energy, the next intonation unit. There's another feature of prosodic breaks that hasn't been discussed much, which is called vocal fry. People maybe know what vocal fry is because it's a stereotypical, it's considered a stereotypical young female West Coast American feature of speech, but we all do that, and especially at word boundaries. For those who don't know what vocal fry is, here is a little thing I found on YouTube to explain what that is. And you're going to hear a sentence that they call it normal tone example, and then with vocal fry. But at the normal tone example, there is vocal fry at the end of the unit.
Thank you for considering me for this opportunity.
Now listen to the second that exhibits vocal fry.
Thank you for considering me for this opportunity.
Okay, so I hope you could all notice the creaky voice, right? Vocal fry is this creakiness in the voice, but it's not just something that, you know, a sociolinguistic feature of a specific group of people. This is something we all do, and again, mostly on prosodic breaks. Zooming into the lengthening feature, this is a figure by Mark Lieberman from the head of the Linguistic Data Consortium at the University of Pennsylvania. And I just really like this figure because I think it really shows the prepausal lengthening I was describing. So what Mark did, he took a corpus of speech, one of the corpora they have at the LDC, and he segmented it based on pauses. Okay, so again, no syntax here. Each line here is the pause group, right? All of the words or the intonation unit. And on the y-axis is their duration. The label 1 is words that have a pause right before them and a pause right after. 2 is a group of 2 words. 3 is 3 words, and so on till 15 words. And what stands out here is the lengthening. The y-axis is the duration. It doesn't matter the number of words, right? Right before a pause, there is this huge lengthening of the word. Again, an average and huge corpus. And it's also really nice to see that there is also a little like dip, right before it. So the word becomes faster 2 words before, and then it becomes slower. So what I did is I took words in the speech of our healthy participants, the older controls that you saw at the beginning, and I took all of the words that came right before a pause and drew their duration, but I also subdivided them into words that were a syntactic boundary and were not a syntactic boundary. And this is important because this is putting together syntax and prosody and looking at it from that angle. And what we see here, first of all, when there is no boundary, we see the prepausal lengthening. Having a pause, I mean, it's a bit hard to see, does make the word longer. Each line here is 1 participant, by the way, and an average over that participant's speech, and the black is the average over all participants. But what we see here is that whether there is a pause or not, a syntactic boundary, and this is a sentence boundary, what I call the sentence, makes the word longer. And this is independent of the number of syllables. I could have normalized it by the number of syllables. Here I'm just showing you the duration per number of syllables for a boundary word in red, which is a word that closes a sentence, the last word in a sentence, and a non-boundary word in cyan. And again, this is a very strong effect. There is a lengthening that is not prepausal or at the end of an intonation unit, but at the end of a sentence, a grammatical syntactic sentence, linguistically defined. So again, I mean, maybe this is not surprising, and I'm not the first one to talk about the fact that syntactic boundaries are marked by prosody. Prosodic cues are part of the reason why they're there is to disambiguate syntactic boundaries, right? So when we look at 2 very similar sentences, when Roger leaves, the house is dark, versus when Roger leaves the house, it's dark, right? You can hear the difference in the way I pronounce the word leaves. And that gave you a clue of whether there is a syntactic closure right after leaves or not, right? When Roger leaves, the house is dark, even without a pause. You heard the lengthening, the reset and pitch. So there is an alignment between prosody and syntax. This, again, should not come as a surprise. So if we look at a string of words, if we think about the strength of the prosodic break, let's say we can take all the prosodic features and somehow compute a score of how strong the syntactic boundary is on that word, we will not be surprised if that will be aligned with the syntactic strength of that word. The syntactic strength is how much that word closes a syntactic phrase when, let's say, sentence is the strongest. So these two are aligned. This is what we expect, but the alignment is not perfect. So here I'm showing a paper, a great paper that was published last year by Giulio Degano from the group of Nali Golestani. The name of the paper is Speech-Prosody Enhances the Neural Processing of Syntax, which is a great title. And this is exactly what I was trying to say up until now. We need prosody in order to support the processing of syntax. I'm not going to show you the results, just this figure, where they show that, so the boundaries here is prosodic breaks. Okay, I use the word break to separate it from syntactic boundary. When there is a strong prosodic break in yellow on the right, okay, very often it also closes a phrase boundary, right? But also not. 28% of the times it doesn't. And when we look at weak prosodic boundaries, there are many syntactic phrase boundaries that are not marked prosodically. And this is also something that there is, there is evidence that pauses are not that consistent in spontaneous speech. And in general, spontaneous speech, because it is unplanned, it is on the fly, then the alignment between the prosodic strength and the syntactic strength, or the syntactic, how much it closes a phrase, decreases. And this is actually good news for us because this opens a window of opportunities. Because let's say, and this is something that in my lab we are currently doing, okay, it's not a let's say, this is something we are currently working on, generating tools to give us scores for the syntactic strength and the prosodic strength. And since they are less aligned in spontaneous speech, then we can use these in order to predict, for example, neural signals. We can use them as regressors. And then we can separate the prosody from the syntax in brain processing and perhaps discover how much the prosody cues for sentence segmentation in order to know where one sentence ends and another begins, but also where syntax does that. Because when we hear speech, when we process language, we also build up partial representations all the time that serve as predictive cues as well. So we have two sets of cues, one which comes with the input, right? The prosody is acoustic in nature, and one which requires higher level analysis from frontal regions. So the other, right, is more top down. We can ask how they interact in the brain. Another thing we can do, right, to move from comprehension back to production is to say, wait, this is not just a regressor, it is a feature of a speaker. We can quantify the sequence, right? We can have a sequence of prosodic scores and syntactic scores per speech sample and talk about how well they correlate with each other. And this will give us an indication of how well the system is synchronized, the syntax and prosody. Now, the big news here is that we are currently doing that without segmenting the transcriptions into sentences. We are working with large language models that are currently fine-tuned in order to provide us these scores without sentence segmentation. And again, this is very, ongoing right now, and I'm very happy to give more information and details to anyone who is interested. But right now for this talk, I just want you to have this, to get this idea that we have these two sets of cues for sentence segmentation, and they are processed in different places in the brain, and they can be an indicator of the speaker's alignment of syntax and prosody. And why do I think that this is, this alignment is something that may be different between groups? So here's another paper, not of my own, and with this I will end, and I'll open it to questions. This is a paper from 20 years ago, and they looked at how older and younger adults process sentences like these. So this is a sentence that I gave you before: when Roger leaves, the house is dark, versus when Roger leaves the house, it's dark. So here the slash is the syntactic boundary, and the percent sign is the prosodic break. And in this case, they are in the same position, so they are cooperating. What they did in this paper, they looked at cases in which prosody is misaligned with syntax. So prosody and syntax are in conflict. There's a conflicting cue. So for the early syntactic closure, this will sound something like, when Roger leaves, the house is dark. And for the late syntactic closure, it will be, when Roger leaves, the house, it's dark. Now, the participants in this experiment had to recall the sentence as accurately as possible, and according to the syntax. That was the instruction that they were given. So these are the results that I recreated this figure. We're not, the early closure, late closure is less of interest here. I want us to focus on the conflicting sentences. So on the y-axis is the percentage of the participants who reinterpreted or recalled the sentence based on the syntax. Okay, so the prosody was conflicting, and they weren't fooled by the prosody if they were at 100%. So we can see that in the cooperating conditions, right, those that are shaded they're close to ceiling, but in both groups, the young adults and the old adults, right, they reinterpret the sentence closer to what the prosody suggests in conflict with the syntax, but the older adult do it more. Okay, so the older adults, they rely more on prosodic cues. This is the interpretation, my interpretation of this paper. They have these two sets of cues, and they are more inclined to reinterpret what they hear based on what the prosody provides them and not the syntax. And this fits very well with the hypothesis that prosodic cues are bottom up, right? They come with the input, while the syntactic cues for sentence segmentation are top down. They require some more high-level analysis, which declines faster at older ages. So with this, I want to summarize the points that we saw and what I'm working on. So first of all, different groups may rely to different extents on prosody versus syntax when they have to pre-process speech. And returning to PPA, right? I started with PPA. Currently, I'm not working with PPA, but the tools that I'm building now, I believe, will be very helpful in studying pathological speech, because maybe this new measure will be sensitive to syntactic planning that is impaired in speech pathology. And unlike my syntactic complexity score, that required sentence segmentation, which was a huge challenge. Here, the expectation is that we will not need that. Now, another advantage that is very obvious but still worth mentioning is we're using here continuous predictors, right? We're generating regressors that are continuous predictors that, unlike the paper that I showed you with the conflicting syntax, the conflicting prosody and syntax, this enables us to study speech in its natural form, not, generating artificial sentences that are not naturalistic in any way. So we can study speech in naturalistic, ecological designs, right? This is the buzzword now. As I said, it's more like I gave you like a broad picture of my thoughts and ideas based on working on pathological speech and production and based on results from cognitive neuroscience and comprehension. And they really, interact, at least in my mind. And I'm very happy to share more of what I do if you have any questions. But this is a broad picture of how, I shifted into studying pre-processing of speech. And this really opens a window to do many very cool things. So I'd like, first of all, to, went over many different studies here and many people were involved in these studies from the University of Pennsylvania and the LDC and the University of Oldenburg and Bar-Ilan University. And also I'd like to thank my research assistants at my lab who work on annotating a lot of speech data in order to develop tools to quantify the syntax-prosody alignment, and other collaborators with whom I'm working on these things. And I'm happy to answer any questions. I'm right in time, right? 50 minutes. Should I stop sharing? What's the protocol now?
You can keep it up if you want.
Okay.
Something. Thank you so much. This was a really wonderful talk. We've got a few minutes for questions. If we have any questions in the room, we have any online. Yes, we got a question in the room.
Hi, thank you for the great talk. So we actually collect the cookie theft data in our healthy aging study here. And I had a couple questions actually about kind of your approach. And first is, how automatic is this? How automatic is it? Because I know we transcribe and we code, and sometimes when you say automatic, I think this is going to be like a magic and we can just feed it and it works. How automatic is it? And the second question is, do you think that by removing fillers and removing some of that content that you're actually could be losing valuable information that is important for cognition and for aging and even for PPA?
Okay, great questions. I'll start with the second one, and then maybe you'll have to remind me of the first one because my short-term memory is So yes, there are studies that show that the use of fillers is a signal. It's not noise. So of course it is. And it's very important to not, I don't want to throw them away because they're not important. But if I study syntax, then they are interfering. That's the only thing. They can be part of the signal with the syntax. But if I want to, for example, extract the number of embedded clauses, the fact that there are fillers there is problematic because I can't, I'm risking myself of getting a wrong syntactic parse if these words are in there, which leads me to the answer to the first question, which apparently I did not forget, what's automatic there? So syntactic parsing is automatic. These tools were trained on well-formed sentences that do not have fillers, that have a start and an end. And this was fully automatic, the syntactic part. What was not automatic? The transcription was done by LDC. It was semi-automatic. And the pre-processing, the cleanup was manual. I tried to use automatic tools. I tried to segment the speech samples using Whisper, I built a classifier to segment it into sentences. I also had to remove the fillers because the non-lexical fillers are not always that easy to identify in order to remove them. Again, not because they're not signal, but because I can't have them there for the parsing step. And that was manual. And that was a lot of work. So in the paper that I cited from 2023, there is a comparison between the manual, when everything is automatic using Whisper to transcribe, and then using the Whisper segmentation, because Whisper also adds periods at the end of sentences, to manual segmentation and cleanup. And there is still work to do there. That's why I'm currently after, syntactic assessments that do not necessarily require this comprehension, this segmentation, and maybe, better ways to segment it. If we understand how the brain does it, maybe we can do better.
Perfect. Thank you so much. Any other questions from the room?
One more follow-up.
Yeah.
So my question is actually a little bit off your topic, but in collecting data, I noticed like a lot of, with this picture in particular, a lot of our younger population have a lot of commentary that they add, or like subcategories of like, oh, well, I wouldn't be wearing that dress while I was doing the dishes. Or like, she's a little overdressed for, they add a lot of like subtext, and how does that affect like the semantic interaction with the picture? How does that inform the syntax that you're getting out of that content, right? So instead of just saying the woman is doing the dishes, the woman who, oh my goodness, I wouldn't be wearing that dress or heels. Oh, and she's doing the dishes, right? So how do you see the semantics and the syntax kind of interacting?
Yeah, so the other side of the coin here is that the older people do not use very, elaborative sentences. And this is indeed my experience. Actually, from the other side, I noticed that older people, this is one of the reasons why I don't like really this picture, is that they tend to not always form full sentences. And even if they do, they just think that they need to I don't know, just list everything that they see. So you'll find sentences more often with the older people. I see a girl, I see a boy, I see a plate, I see a sink. I'm a bit exaggerating, but I fear that oversimplifies their syntax. It doesn't really probe their full syntactic capacity. So this is just one reason why I feel that the cookie theft picture is not good enough. And I know that now the world has moved to other pictures, but then, it's when you collect rare diseases, it means that you have to give up over like 20 years of data collection. I don't remember that like with our younger adults, they gave that many commentaries, but they definitely used like more full sentences with and I think that, their syntax is richer because they were maybe feeling more comfortable providing more information other than I see a sink, I see a mother washing dishes, I see a child on a stool. Yeah. So I have a similar impression, but from the other side of the coin.
Thank you. If you can't see me, but I'm nodding in agreement.
Okay. Who am I talking with?
This is Sarah Newman-Norland.
Okay. Hi. Nice to meet you.
Hi, Jesse.
All right. Maybe I can end with one quick question. We got about a minute. I wanted to really ask you to elaborate on sort of a question that you posed at the end of your talk, whether prosodic cueing for syntactic boundaries, we should expect it to differ between populations. And in that case, pathological populations, in particular, people with degenerative brain damage or post-stroke aphasia, those populations, do you think it would be helpful, and how, maybe?
So the clue for me is that paper that I showed you that gives evidence that older people rely more on prosody, which is something that I intend to validate using naturalistic stimuli. And if we find that in older people, that already makes me think that we may find that in pathological cases as well. And again, this is the hypothesis that prosody is comes with the acoustic input. So it's more temporal, and the syntactic cues are more top-down, and we should see that in production as well. So the whole system will be less synced in a way. This is the hypothesis. Now, why is this helpful? Because if I can score the speech with the models that we're now training without segmenting them into sentences and without removing fillers, that will be very convenient, to say the least, because the syntactic complexity score that I showed, or any syntactic complexity, there are many metrics to quantify syntax. They always require a sentence, and they always require a clean sentence. And therefore, it will always be challenging unless we find an easy, automatic way to pre-process the data for these algorithms. So what I'm suggesting is a way to measure. It's not precisely syntactic complexity, it's the syntax-prosody interface, but it has to do with syntax and the balance between top-down and bottom-up cues, hopefully without the need to pre-process the whole speech. So that's the benefit.
All right, that's great. Thank you so much. And that brings us to the end of today's talk. Thank you once more, Dr. Agmon, for a wonderful talk. And thanks to our online and in-person audience for watching and asking questions. All right, we will see you again in 2 weeks' time. Thank you so much.
Thank you so much. Bye, everyone.